Color for the invitation. Okay, yeah, and yes, just click OK. Right, and thanks for the invitation and a nice introduction. And really glad to introducing our work and here and actually I feel really excited. Like we have such a center and focus on the molecular probe for the mechanical detection for mechanical technology, and I feel this is indeed a need and to promote this field. And so, yeah, actually, like when I prepared the slide, I realized like this is for the first time I use the whole 45 minutes or 1 hour to talk only about mechanical force, because often I was invited for give the other half of the group to talk more about afterwards stuff. Right? But I'm really glad like we can talk more on this DNA for this mechanical things. And so basically, this is the title and what I called. And so the focus of the today's seminaries will be more on the cell junctions, and I will tell you like how we are developing different kind of probes or maybe also some regulators and to map or to potentially regulate these intercellular forces. And so as Carlos said, already introduced, and so basically like my lab or like my training is mostly focused on the DNA and RNA or nucleic acid based probe design. And the things which is really draw me to this field is because as a chemist, and we think the DNA and RNA really have very precise self-assembly properties, right? So it's a kind of biopolymer and biocompatible. It's a right material to study the cells or to study the interface between chemistry and biology. And also potentially this DNRA can be genetically encoded, and you can use it as a part of the central dogma, and you can use it for some potential programmable regulations or detection. So that is the things like really drive me to this field. And so, yeah, I have to mention maybe one of my first work that time was we tried using light to control things we call DNA walker. And that time it's hard to convince Wei Hong to say like why I need to do such kind of job. And because we were driven more like by the disease, by some of the cell studies, but that time it's more focused on the molecular probe design. And uh, I would say that time we have no clear reason to do that, and we just feel it's interesting. Like you can use different things, you can drive in the motion of a molecule of like a DNA. And later, I hopefully I can convince Wei Hong and convince all of you to say like indeed this DNA probe, and by combining with some of molecular motion, by combining with some of the mechanical features or these physical features, and it can be used to study cells. It could be used for some potential understanding of cell biology or for disease stage as well. And so another part of the things we have been really interested in is this DNRA not only can have self-assembly features, not only based on their ATCG or UCG base pairing, but also they can have very different functions. For example, they can be used as enzymes themselves, or they can be used for recognition of different metabolites of proteins. And so we will not focus on this part of the work today, and but we will see how things are going, right? And so basically, like in my lab, and we have two subgroup, and the first subgroup is focused on we call the genetically encodable RNA-based sensors. And so basically, these are some RNA aptamers, and which can be genetically encoded, and you can the cells like can using this plasmid, can using this gene, and to synthesize different functional RNAs. And then you can use it for some cellular imaging of RNAs or metabolites or proteins. And so, yeah, I will not talk about this part of the work today. And so the things I will focus more will be on the cell membrane. And as I mentioned, we will talk mostly on about how we can using this different lipid modified DNA probe and to study the mechanical force on the cell membrane, and also like on this cell surface. And we have some other ongoing interest. For example, like we try to image or regulate different molecular interactions. and they're not between the cells, but maybe like along the same cell membrane. And so those are some of the work like we have been interested in right now. And so what I will start with is I will start with how this lipid DNA work on the cell membrane. And so for me, actually, like we enter this lipid DNA field by chance. And when I was a PhD student in Wei Hong's lab, and that's how like we are interested in some of gene delivery or DNA RNA deliveries. And one technique and we saw is like you can modify a lipid and with a DNA. And so that's how like we modify a sulfur with a lipid DNA. And what we found is you can have this fluorescent signal on the cell membrane. And so after I established a lab, and I kind of think about what direction I should go. And I kind of feel like this is a very interesting feature, and because you can have a very easy way and you can have very efficient method, and you can modify the live cell surface and with some functional DNA probe. And so basically, like I convinced one of my first bunch of PhD students, Yusuf, and try to synthesize different kinds of lipid probe. And so basically here, like most often, people in the field will be using cholesterol DNA because it's commercially available, and also like based on the modification efficiencies, it can highly efficiently modify it onto the cell surface. And but indeed, you can tune the different hydrophobicity, you can tune the chain lines, you can change and their saturations, and you can change the chain numbers, and basically you can tune and this membrane modification efficacy or efficiency. And that time, like what we found is like basically again, like you can do a very simple and a very effective way of a modification. And in a way, this is a very general approach. Sometimes general is not good because you cannot get targeted modification, and sometimes if you want to get only modification on the T cell, only modification on the B cell, it's hard. And but um, there's some other strategies and potentially we can solve it, but it's again this is a lipid DNA probe is kind of general, or I would say in such a way. And so basically, like I would like to highlight some of the work and the user has done, and I think this is very important to understand how this DNA lipid can anchor and what kind of kinetic pathways, what kind of destinations, and we can imagine. And what we found is basically like by changing different lipid lengths, by changing also the DNA sequence and different environmental properties, and actually like we found you can tune like how much of the DNA you modify and how quick you can modify, and also like for example how much of the DNA and you can anchor onto it. And but meanwhile, like some of the interesting thing we found is, for example, like if you change the hydrophobicity of the lipid. And at the beginning, we thought like maybe we need to have a really hydrophobic probe and to get a more efficient modification. And it turns out like basically the both kinetic and efficiency wise, and it's not to say like the more hydrophobic the better. And we found you have to balance the micelle formation, the in vitro monomer versus the potential aggregates and in the solution, or like you want to tune the CMC values of this lipid DNA conjugate, and in order to have a efficient modification. So in that way, the simple conclusion is like cholesterol will be still very most efficiently, and you can modify onto the cell surface. And but later I will say some of indeed some of the limitations and this lipid DNA cholesterol modified DNA probe still are experiencing. And another thing is like we found like from the kinetic wise, and we can have this lipid DNA probe, and definitely it's not that stable. And over time, and they can still like go through endocytosis. They can still like entering cells, and then like and you cannot use it anymore for the cell membrane status. And what we found is like basically in many cases, and within around 2 to 4 hours, and if you don't have actual probe and actually like this cholesterol modified probe, and will basically mostly will be getting internalized. And here is the EP just in short for the actual probe. And but there's a couple of ways, and you can try to elongate and the membrane signals. And the simple way is like if you have actual probe and basically like you have this continuous equilibrium. You have a new probe anchoring, and then the old probe get internalized. So that is one way. And another is like if and you care really a lot on how many probe will be stay on the cell surface. And we study different kind of endocytosis inhibitors, and we found one of the inhibitor and this Philippine actually has the most efficient and in promoting the membrane we call it membrane retention or membrane persistence of the probe. And so that is another way if you want to get a day long of incubation. And so that is the trick you can play. And so after talking that, and basically like what we also found is on the cell membrane, and you can have very still very dynamic interaction ongoing. So that time, like one of the things we were wondering is once you modify a DNA onto the lipid, and how much of the lipid property will be changed, and whether like the lipid cannot move freely on the cell surface because we saw that at that time, like the DNA, the molecular weight is much larger compared to the lipid. And but based on the things we measure based on the FRAP or based on FCS, it actually turned out like this DNA modification, even though their molecular weight is much bigger than the cholesterol or compared to our some lipids, and actually like their membrane diffusion rate is very similar and before and after you modify with the DNA. And so that is very interesting. And we found like in this way, we can have still have from the lateral membrane case and can still have a free diffusion probe. And I think that is also a basis for the later try to use it as a way to measure the intercellular forces. And another thing, like we also characterize on the cell surface is also to see how and this lipid modification will change the membrane different interactions or membrane dynamics. And just to show you like one set of data and we found very interesting is, for example, we at the beginning we thought about to study different membrane dynamic interaction. Here we talk about is the safe way of interaction. So two proteins or two lipids on the same cell surface. And in that way, like we found in the traditional way to image and gold standard is still Fred probe. And but however, like it can only use for some really stable interactions. And what we try to do is like whether we can by anchor a DNA probe, and whether we can tune this membrane binding affinity, and whether we can tune this membrane interaction time. And indeed, like if we can increasing the length of DNA. So for example, here on the y-axis, what you see is we change the DNA probe length from 6 base pair and up to around 10 or 15 base pair. And indeed, like once you get more of you get additional donor probe, or if you get a longer piece of DNA duplex, and you can have a tunable way of this membrane interactions as measured by this Fred or measured based on some of fluorescence signals. And so after all this calculation, and we realized like there could be something like we can use in this probe for on the live cell surface. And so basically, like I the things that drive me to this field of mechanical probe engineering was when I talked with my one of my friend, Professor Mingxu at mechanical engineering department at UMass. And actually, like that time, like when I started my lab in 2016, and I found like probably I will focus more on the tool development. So the first step is I should know what kind of a challenge, what kind of limitation in different field. So when I talked with Yubing, and he was able to convince me like there is a need and to detecting to measure this intercellular forces. And so this is a review like Yubing has previously published. And what he showed me is basically like there is a couple of ways and you can measure and the cell and the matrix interactions and including this DNA probe, including like these molecular probes. And there are a couple of ways you can control and you can study how the shear stress will changing the cell behavior, how to change the tissue or different development process. And but what is much less studied is the forces and between the cells. And at that moment, and I think there is a couple ways, and the way people do is like based on the cell matrix interaction and try to derive and what it will be the force between the cells. And with assumption, because the cell will experiencing a net zero force in order to maintain their current stage. And however, like in that way, you cannot quantify the molecular level of forces. You can likely study like the whole cell level of this force. And there are some other ways, and you can synthesize a bow tie shaped engineering device. And also that time, like there are some genetically encoded fluorescent protein sensors, and that has been used to study, for example, the calculation forces. And I will still like compare to these things. And when what even convinced me is nothing is perfect, and there's still like many room can be done, especially at the intercellular force range. For example, how we can make it to be already highly sensitive detection, how we increasing the signal to background ratios, and I think many of the things like the center of the probe molecular technology are doing right now, and I think they can be potentially applied onto these intercellular junctions as well. And so basically, like at that time, like when you've been introduced this problem to me, and I immediately thought about the early work I saw from the Selecta lab, and tried to using this DNA hairpin probe and to measure and this mechanical force. And so basically, like the things, and I think this side audience already quite familiar with is like we can have this DNA hairpin probe and which can have a different sulfur and quencher modified. And that like by changing different ligand, and you can study their ligand experiencing forces. And what our contribution is like, we thought about to put a lipid anchor there, and we thought about like we can put it down to the live cell membrane instead of on the matrix. So in this way, the lipid will basically like recruit or drive this DNA hairpin probe and to the cell-cell junction. And so what is the scheme showing is like basically it's an intercellular junction regions. And you have one cell on the top, you have another cell at the bottom. And so based on this design, what we found is maybe like we just simply try some of integrin ligand like RGD or some of cadherin, and which is the things which is driving this epithelial cell to be come together and to see whether we can use it as a demonstration of this concept. And so basically, like that time we have already attended the postdoc, Professor Dr. Bin Zhao. And so basically, like what Bin has able to achieve is, and again, like we first modify this ligand and to the DNA probe, and again, like we modify a sulfur and a quencher here together, and we just put it under microscope. And after around 15 minutes of incubation of the probe. And so what we found, indeed, you can use it to study different level force. And that time, like the things really pushing us to think about this will be a good way and to detecting interstellar forces. Again, is already known and based on the early contribution from the center, like we can using this DNA hairpin to have a tunable the force range, and you can have this piconewton level force, and which is the similar to the cell to the matrix, but also like between the intercellular region. And in many cases, I would say not in all the cases, and in many cases, and this force will be still in this range. And so another thing is like definitely you can change the DNA sequence, the DNA length, and later I will show you some about the design we have. And you can also have achieve and different kind of features and for example, improving the multicolor or improving the sensitivity or for some of the simultaneous or some of the different kinds of detection purpose. And so early, like what we tried is the most exciting things that come from again from the integrin, right? So the integrin really have a strong force and really easy to visualize. And so we simply put a previously developed probe and which is around 4 piconewton, I believe, and for the threshold force, and we just modified RGD there. And that time I don't quite know the much difference between different subfamily of integrins and I just say, okay, that is what the people do, what Kala highlight is already doing, and let's try the same thing. And so basically, that is a way like we try to incorporate this probe, and so we'll be studying these 3D cells, and we found it's quite interesting. And based on the different molecular features, based on the cell phenotype, and you can indeed experiencing different level of intensity of this light. And so basically, like at that time, we found that you have depends on the surface size, depends on what kind of whether it's a nearby interaction or remote interaction, and the interaction pattern itself is quite different, right? So that drives me like very excited, and I feel like this is indeed something quite interesting. This is indeed something, and so the field likely will need. And so basically that time, like after we finished the first work, and we started thinking about like what kind of direction and we need to go. And so for the rest of the time, and what I will show you is some of the effort and we have been working on to solve part of the problem. And so I would say first of all, like and how we can quantify these forces. And that one is not so challenged, I would say, even though like there is indeed many things you need to optimize. And but in terms of other things, is sometimes, for example, like how you can achieve the multiplex detection, and it takes us some time to figure out. And so there's still some of ongoing challenge, for example, especially for this lipid DNA probe. And so how you can achieve a more reliable and also like for a longer term of detection and longer than an hour or two. So that is still some of the, we have to play with some of the trick, and we have to play with some of the optimization I will show. And so similarly, we also start about like how we can apply this probe and not only for imaging, but also use it to understand the different cell features. And to be honest, like for the early work and for the early version of the work, and when we try to study different real understudied the system, and we face still several challenge. And I think yeah, Cheng probably knows some of them. And so and also like when we sometimes we realize like when we share the probe and to the whole community, and so it's quite different from the RA side. And for the other half of the group, and what we share is often a plasmid. And the people working in the biology, they really get quite familiar with how to handle the plasmid. They really how to familiar like how you can use it for their imaging. But when we like deliver the DNA probe and even the simple annealing and even like we assemble 3 probe, and they still not quite well accepted. And so yeah, I think some of the work and the center is doing likely hopefully can also solve those problems in how to broadly advertise and making the real community of the mechanobiology field people can use it instead of limited to a handful of the groups here. And so yeah, but it's still something like we are trying to do now. And I will not say that is quite successful, but I will show you some of the work we have been doing. Okay. And so I will start to say like by showing this quantification things. Right? And again, like the one thing like I think this DNA probe will be quite different from the fluorescent protein or peptide-based probe. Is it's more like a digital probe. They have a really narrow the force detector range, and in this way, like it may not be great to detecting like what is indeed what is the level of force you have, whether it's a 4 kilonewton, whether it's 6, or whether it's 8, is probably harder. And I think right now we have some better strategies. And by that time, like so digital will be great in terms of you can quantify the number of probe being opened. You can calculate based on each pixel and how many probe are open, how many probe are closed. And from the quantification, and I mean more about quantify how to convert from image to the percentage of probe, percentage of pixels being activated. And so basically at that time, like we have 2 strategies and for this quantification. The first one is based on this ratio metric image. And again, the one difference and from the matrix modified DNA versus cell membrane is cell membrane have a more heterogeneity. So basically, different cells, different region of the cells, and they will have a different amount of DNA probe. And so in this way, like at the time, you have to have a reference probe to give you this information regarding in each pixel how many DNA probe are there. And so in this way, like we can base on the probe and which is the dye which is used to detecting force and divided by this reference and to calculate how many percentage and of the probe was able to be activated. And that time, like also because and for each DNA hairpin, and you can only tell whether it's above or below 4 piconewton and something like that. And so basically, like we thought about maybe we just put a chain of DNA hairpin there, and in this way, you have one DNA hairpin to detecting the range from 2 to 6 piconewton, and the other one maybe detecting from 6 to 10. Right, so that is another thing we call it a DNA meter. And so basically here, like what we have shown is, for example, we're using the cadherin, the E cadherin as an example. And if we modify the yellow, like the sensory as the reference, and we're using green, I forgot it's a FAM or likely the FAM, and as a force, as a probe to detecting the low threshold force, and using I think it's sci-fi and to detecting the red color for the high level force. And in this way, based on the ratio between either the green to yellow or red to yellow, and you can quantify like percentage of pixels undergoing different level of forces. And as a control, if we don't have ECAD and we don't have the ligand to be recognized by the natural cadherin, and almost all the probe and are in the inactive form. So basically, that is tell us like indeed is a force-induced opening of this DNA hairpin. And you can also convert this percentage pixels into percentage probe, and by normalizing like what is the number of probes in each pixel. And so again, like those is kind of work early on, like when we try to do this ratiometric image and base the quantification. And later, like when we try to use it, and at the time, like we thought about some of the ways and to overcome one of the limitation, as I mentioned, is for some of longer term of studies. And again, like when we try to image the probe, and many case, and we had to finish the imaging within 2 hours, because the longer that you will start to see the probe internalization. So most of the cholesterol DNA will be drive, go to the endocytosis, and so that will be a limitation. And at that time, like, but we still we want to study some of long-term interactions. For example, like the system we chose is this cell aggregation. And what we are trying to do is like we want to see, and if you grow a layer of epithelial cells on the surface, and if you have a PDMS and to block the motion, and if at time zero you take off, you peel off this PDMS, and can you study like during the cell aggregation and what is the force distribution between the cells? And that's how like the trick we did is basically like we can adding the probe, and we found like you can after 3 hours often, and the first batch of probe will be already gone, and many of the things and can be degraded. And that time we didn't try to improve the stability of the DNA probe. And the good thing for that is like you can just adding the new batch of probe, and so you can do another round of detection. So basically you can do 24-hour, you can do 12-hour, even longer, and so basically you can use it for some of longer term of studies of imaging. But again, you cannot do the continuous detection using the same batch of probe. And so that time, like when we studying this cell aggregation, again, like to time zero, we adding the first batch of probe, and we take the image. And then like after 6 hour, 12-hour, 24 hours, and we take a bunch of other image. And what is interesting we found is like, so initially like you have the edge of the cells around here, and after 12 hours, and so basically the cells already aggregate a lot. And so in that time, what we found is like if you quantify the distribution or like the number of these active pixels, and actually there is follow a linear increase over the distance from the leading edge. And actually, that is time I thought I thought about the opposite. I thought maybe the cell moving forward will be applying the tensor force, but it turned out to be the opposite. And so the cell don't want to move, and so the cell which is far away try to holding back like other cells. And that is the one way like showing indeed you can using this probe, and you can study the spatial distribution. You can also study some temporal change and how this during this process and what kind of intercellular forces are happening. And so that is the first way, like we do the quantification based on the ratiometric detection. The second way, and we did that time, is we also think about the fluorescent lifetime. And again, the lifetime is a great way, and because it's concentration independent. And as I mentioned, like one of the big challenge of our lipid DNA probe is like on the cell membrane, and at different location, you have a different amount of DNA probe. And if you do the traditional way of detection and we changing the probe concentration, and you can see like by adding more probe and you have a higher fluorescence. And but if you do a lifetime based detection, and basically like the fluorescent lifetime of the probe will be the same and independent of the probe concentration. So in this way, the big advantage is you don't need to add in the reference probe. So you don't need to add in the reference dye. And so that saves us the space for this fluorescence, right? Because again, the fluorophoros and you have spectrum overlap, and you cannot have so many fluorophore to be imaged at the same time. And so in this way, like we try to use it for the multiplex detection. And the good thing is like based on the flame and based on also the traditional ratiometric image, and you can have a nice correlation in terms of their the fluorescent signal. I think this is based on the ECAT signals. And so again, like after we confirm, like indeed you can using and this probe to detecting both, for example, the E-cadherin and N-cadherin, and we applied this probe to study one phenomena, which is the EMT, the epithelial to mesenchymal transition. And the reason we choose this system is because during this process, and different intercellular forces or different partners of intercellular forces are experiencing, including the E-cadherin, N-cadherin, integrin, and several others. And so basically, like in this way, and we are able to quantify the same. We can quantify the same time the ECAD and kind of force distributions, and after different inducer treatment, after different time of this EMT inducing, and so basically, like again, like that time, like we are able to quantify or detecting like different triggers, and they do have a different distributions or different functions in terms of this activating of intercellular forces. And so basically, like this is the second way, and we try to do the quantification of the forces. And so later, similarly, like we also think about what whether this kind of this cadherin forces will be also depends on the cell environment. And one big thing is that we thought about is instead of from the 2D cells, and whether we can move it to a more 3D and cell environment. And so the simple thing like that time we thought about it will be some of a spheroid or like organoid. And so basically, like what we try to do is we want to first characterize whether this lipid DNA probe can indeed anchor in the 3D cells. And so what we did is like basically we modify so this cholesterol and with itodye and with the DNA, we just incubate for different time. And then we're using confocal microscope and quantify different layers of the cells. And we want to see like how deep and this probe can go, and whether it's a concentration of the probe dependent, and whether it's the incubation time dependent. And indeed, like we found this probe can, they do have their limitation in terms of how deep they can modify. And for some of the big spheroid, if it's larger than around 300 or 500 microns, and this probe cannot go to the center, the core of that. and even like we incubate for much longer time. And but still like so current probe are still able to at least penetrate around the 10 layers of cells, and you can use it to quantify some of this fluorescence, quantify some of the intracellular forces. And again, like what I show you here is the E-cadherin force distributions. And inside of this spheroid, I think this is MCF7 derived spheroid. And so basically, like you can see, like again, the center core of this spheroid, you cannot see the fluorescence, likely again due to the penetration of the DNA probe. And assume this is after 1 hour of the incubation, and then like we can using this ratiometric signals and to study how and this cadherin distribution, cadherin forces can be distributed and in the 3D and from the different layers of the cells. And so yeah, basically that is the things like as I mentioned that we early on try to study different quantitative measurements. And so the other features and we have been also experiencing is also like, and as I mentioned, it could be including the sensitivity of the probe, or like how you detecting potentially some of more transient or some of a low level forces. And again, like the good thing is like on the cell matrix inductions and many of the DNA probe has been engineered and again, like inspired by some of the earlier pioneer work and in the field. And we thought about like whether we can use it for some of the intercellular force detection as well. And so here I will show you first the two type of things, and we try to improving the sensitivity of this probe. And so what Maruli has been doing is one thing is we are using the CRISPR, and that is what Maruli's training background, and to see how whether this one can be similarly to the cell matrix to improve the sensitivity, whether we can use it for this intercellular force detection as well. And so again, here, like we focus on the fluorescence imaging. And what we did is we labeled this cholesterol DNA probe. And then like we also having this Cas12a and in the system together with a guide RNA. And what happens is like before you have the tensile force to open this guide RNA recognition site, and basically like you cannot have this Cas12a to be activated. And once you have this exposure of this red domain to recognize by the guide RNA, and in this way you have two type of cleavage. And one is the cis cleavage of the same probe, and the second is a trans cleavage of the nearby probe. And what we modified is like basically we have a different Frother quencher modified DNA probe, and also with a cholesterol anchor. And this probe will be freely diffused on the cell surface. So in this way, our goal is like, can you based on the ratio between this cis-cleaved size array and this trans-cleaved fan, and to detecting or to monitor these intercellular interactions. And so again, like here, the cis cleavage will making the cell surface size rate signal to be reduced, and will making this cell membrane fan signal to be increased. So this is again like to give you the handout here. And here is what is the result showing. And so uh, we we just showing here, like for example, you again, if you're using the integrin IgD as example, and so if you have this integrin induced cleavage, and so I think this after 1 hour of incubation, I think the shortest time you can detect it right now is around half an hour. And so after this incubation, basically the size rate signal indeed can be decreased, and then the FAM signal will be activated on the cell surface. And but if you don't have integrin, and so basically like you can see the sensory signal will be still there, and the FAM is still under the quenched condition. So basically in this way, using the FAM to the sensory ratio, and you are able to, and to comparing like to study this intercellular forces, right? So compared to the original hairpin-based probe, and indeed this one will have several fold of improvement in the sensitivity. And another thing like we have been also doing, also feel very excited is about this again like some of the previous design, like you can using this another kind of probe and to making this a reversible DNA hairpin probe into more irreversible way of fluorescence generation. And so basically, like the things I want to say here is when you got the traditional DNA hairpin probe, and once you got the force-induced opening, and if the force is released, and this hairpin can be refolded, and then the signal will be decreased. So there is a couple of things and we try to improve. And the first is like we want to improve the signal to noise ratio. And the second is like we try to use it as a way you can actually do for some long term of a force detection. So here is how it works. And so again, like the things we added, and Priyanka is basically like Priyanka adding this log, we call locker DNA strand, and you can modify your fluffer. And so I think again, similar thing has been demonstrated on the between the cell matrix. And so again, the locker strand cannot hybridize with the DNA hairpin, and so only after you have this force-induced open DNA hairpin, and then like you can have the locker strand to hybridize with this hairpin. And so at the beginning, I can tell you like I thought that this would be very straightforward, and because it has been demonstrated on the cell metrics, and so when we tried using the same DNA sequence on the cell surface, and it doesn't work that well, and we have to optimize like different probe, and we have to redesign, re-engineer, re-optimize, and you still don't know what happens. And but I think right now it's good. Like we are able to get this logger sequence, and which can only function and after you have this force-induced opening of this DNA hairpin. And so again, the reason we think this probe will be very useful and for these intercellular studies is first of all like you can convert this transient force in want and into this accumulated signal change. And so if you have some of short-lived forces, and that should be a good way to detect and with a higher signal to noise ratio. And the second is like a big disadvantage of this cholesterol or lipid modified DNA probe is after 2 to 4 hours, you get this probe being internalized, right? So this is will be a limitation, and if you want to do for a long-term force detection. And the things we found that this probe will be good is because the local DNA and by itself will not internalize. And only once you recognize by the DNA hairpin, and then they can be taken and from the endocytosis into the cells. So in this way, like we can monitoring the local DNA sequence, we can modify the local DNA fluorescence, and so basically like we can try to study like whether after different lengths, different time period of infections, and whether we can study these intercellular forces. And here is the result, right? And so basically, like Priyanka tried a different time of incubation, and here is the result after 24 hours of incubation. And so previously, we cannot really image it because, as you can see, is all the probe, and or at least the most majority of the probe has been internalized. And but the good thing here is basically like the Sci3 channel is used to monitor this logger strand. And the Sci5 is play as a reference and it's always on. And when we quantify like this, and when we compare the Sci3 to Sci5 ratio, and we compare three different conditions, what I show you here is like if you don't have RGD, and so basically, like even though they have some of signal increasement, and but compared to the RGD modified probe, and it is still much smaller. And so in this way, like it's indicating like there is a force induced open of a DNA hairpin, and then like you can making this DNA probe to be internalized more. And the second is like also like you can the another probe we designed is a pre-annealed probe, and again, like if you have a pre-annealed, indeed this locker strand will be more stably and to be hybridized, and they can be internalized together and with DNA hairpins. And so basically, like this is the way, and I think that'll be potentially useful for this intercellular force studies. And again, like as demonstrated before, and so you can using the microscope to study this, and potentially you can also using like cytometry or other ways, and you can do some of the long term or like also the high throughput force detection. And I think that is a big problem for now for the field regarding how and you can detecting these intercellular forces. And so I will not go in detail like for some this part of work. And again, like this just a part of work and the one postdoc and try to develop the first version of a portable electrochemical sensor and which you can measure the force. But that time is only for the cell matrix interactions and or the cell gold surface interaction studies, but not yet for the intercellular force. And I think I will just end up with showing one last story. And more recently, like Merli also have finished another work and say like instead of using this force-induced probe opening for detection, you can also use it for this targeted delivery of the drug. And so basically what Murray has demonstrated is like if you added this doxorubicin and into this DNA harping, and if you have this mechanical force occurring, and you could have this targeted local release of this dox, and this way you can indeed induced. And this targeted treatment of cells, for example, the HeLa cell is known to have this integrin expression. The HEC cell will have very low expression. And if you take a look at this RGD probe, and indeed, like you can induce a more dramatic cell death and compared to this HEC cells. And also like in the a simple mixture of this HeLa and HEC cells, and we can achieve this targeted killing of these HeLa cells rather than the HEC cells. And so basically, like this is just a simple way to demonstrate not only we can using this DA device and for the imaging, but potentially you can also use it for some of a regulation. And so that is again like the things we are quite interested in. And to give a quick summary, and again, like so for the DNA probe and the RNA probe, and again, I think it's very interesting and very precise way. You can have this probe to function dynamically. You can use it to detecting different features. And again, like for this mechanical force, especially on the intercellular mechanical things. And we think the tool development is still have to be ongoing. There's many challenges. And for example, I think in the center, one of the goals is how you can get a real single molecule, really like high resolution detection. And that has at least not yet achieved on the intercellular region. And this many of intercellular forces, for example, like we have been really interested in the notch signaling, but I can tell you like we have a really limited success in this notch detection. And I think one reason probably because their intensity of the force is quite low, and or maybe their interaction is too transient. And so basically, like those are still the things how you can detect this transient low level force. And so that is the things has been still ongoing. Right? Yeah, I do feel like and this whole field and is moving forward. And yeah, I'm really glad like we have the center right now. We have a, pack, a side of people with different training background and try to contribute together. And field. So yeah, with that, I want to acknowledge our collaborators. I mentioned Cheng's name, and yeah, to say like we still owe Cheng a probe detecting these T cell B cell infections. Is yeah yeah after you figure out some of the challenge, and hopefully we can re-enter to tell Cheng like there is indeed something like we can measure more robustly. But that is just our current stage. Current. Yeah, so I think with that, I will be happy to answer some of your questions. Yeah.
Wonderful.
Thank you. Right.
Thank you for sharing. Yeah, this is great. I have a lot of questions myself, but I'm gonna hold back and let others, if you want to raise your hand and add a question or use the chat to ask questions, that would be great. We have about 10 minutes to go through some questions. I see that Cheng, unsurprisingly, Cheng's already raised his hand. So maybe Cheng, you could start, and then after that, we'll go through. There's a question I see in the chat. We'll do that next.
Yep.
Wonderful talk. Really enjoyed it. I have a question regarding the force map that you show in the collective cell migration, where it looks like the fluorescents are much brighter at the rear side of the cell collective, and then in the leading end was not so much, right? So I forgot exactly, was that the coherent probe?
Yeah, right. This is a e-cadherin. Yes, right.
So you one side of the cell would have their own coherent, and you provided on the other cell a probe that with coherent, right?
Yeah, we basically added this e-cad probe just in the
So then basically you are increasing the density of eukaryotes on the cell surface, right? And I wonder this pattern. So people like Jeff Fredberg of Harvard School of Public Health has published using I think it's traction force microscopy that measures both the forces that the cell exert on the bottom surface and also some estimate on the intercellular forces, like what you show. I don't remember they're saying that the rear end has a lower force. It has a higher force than the front end.
Yeah, actually at the beginning, like the reason I said I thought maybe different opposite direction is that time like we do saw controversial data, and so people based on the traction force microscopy and kind of indicating like the leading edge has somehow higher force compared to the far end, but other people like using Jack encoded fluorescent protein, and they try to get a similar result as what we show here for the ECAD. And yeah, so yeah.
So, but you are adding molecules on the surface, right? As
right, we do adding ECAD modified DNA probe.
Right. So because you're now having more adhesion molecules that mediating this, I wonder whether or not that would have an impact on the measurements.
This is a
it's a great question. Yeah,
classic question. Does the probe, interfere with the signal?
Correct. Yeah, it's a great question. Yeah, right. So, so often like you have to be careful here, right? So I think so depends to me. It depends on the level of what is the natural cadherin, how much of the cadherin we added, and so I will say yes, and there will be still some interference, and that time like the reason the part I think is still is if the interference to me the interference will be similar, and so definitely we try to reduce the interference and by using a low concentration probe, but again like you have to have a certain number of probe in order to see the enough fluorescence, and but the things I should say is also is compared to the leading edge or the initial edge. And even though, as Cheng mentioned, it looks like the intensity of the fluorescence may be different. At that time, like we saw the modification of the probe will be have a similar effect. Right.
Thank you.
So let's try to get through the last maybe two questions that are in the chat. One of them I'm going to paraphrase by Sunath Mohan Kumar. I think the question is asking, how do you know that you're measuring interactions between cells rather than between a probe and a receptor on the same cell? And I guess that's a question for the RGD probe. In other words,
correct.
how do you know is it cis versus trans?
Yes, right. So basically, like for the RGD case, and what we did is first of all, like we compare the junction versus other membrane region. So basically, like if you have a knot in the junction, and so we thought like you still likely have a cis interaction, you may have a instead of a trans interaction. And so indeed, like we found the junction, you have a higher signal and compared to the membrane. So that's not a yeah, so that is one evidence. Another thing is like that time we did this molecular simulation to see how whether the flow will be try to lay down on the surface or try to stand up. And I didn't show the data here, but mostly on the lipid membrane. So one thing is like the cell surface will be also like a negative charge, and so the DNA will be standing up and within the angle of 30 degree. And so again, we can yeah, we will not say like there is not a chance of a cis induction, and I think the trans induction will be more likely in terms of one is this geometry, and also the other thing, it could be also relevant to the lipid anchor, which is also we think is kind of free diffuse on the cell surface. And so basically like if I always I also being asked the question like why not modify a protein or some other things which may stay longer on the cell surface, and I thought it's just the proteins maybe like they'll be more steady on the cell surface, but also they will be aggregate slower, and so in that case, if you have a cis interaction and there's force, maybe still will activate more of the probe. But if the lipid is really flexible, moving around, and it sounds like we have a less and chance to be opened by the lateral cis force.
Okay, thank you. So two more questions. Maybe this is a quick one. Do you ever see, this is a question by Maya Verangel, and she's asking if you ever see evidence for shearing.
Yeah, right. So it's a good question, right? So yeah, so basically the shearing, I think there is a likely a chance, yes, right? Because again, like this DNA, we tried the tension, the TGT probe on the cell membrane before, and so if you have an integrin, there's a chance you can have a larger than the 50 or 60 piconewton, and so there's a chance of shearing. The shearing will in our case, and we often try to design the probe to have the quencher and the modifier with cholesterol and the fluffer modifier on the other side. So if there is a shearing, so basically the signal will be released from the cell surface. And so we worry about that, like when we try to measure the integrin. But later when we study cadherin, we study the notch, and I think that chance is much less because those kind of force are very small, right? So it's not enough to take off the cholesterol, not enough to unzip the DNA hierarchy.
Okay, great, thank you. And then lastly, Brent Hoffman is asking, essentially, endogenous cadherin is linked in indirect ways to the cytoskeleton, but your sensor is not. How does that change interpretation, or does it change anything in your case? How different do you think it is than the physiological case?
Yeah, it's a great question, right? So yeah, often what I think is, for example, like in terms of a Jack encoded probe, right? So that would be great in a way, like you are kind of a mimic what occurred in nature, and how how their interaction pattern looks like, and but you often have a limitation regarding whether it will interfere with the natural structure of these proteins and how big it is conjugate the probe is. And so for us, I think the one limitation, yes, is here, and this the DNA hairpin is the DNA probe is modified from a different mechanism as how this natural cadherin integrin work, and but still, like we think about the receptor side is still the endogenous ones. So we are measuring like what is the amount of a ligand experienced at that side of the cells, and the force still generated by the cell itself, and we only detecting and the force being experienced by the probe. Right? So that is how in general work. Yeah.
All right. Well, thank you so much. We're a few minutes over 1, I guess, local time here. Thank you again for the seminar, and thank you everybody for attending and asking questions. And we look forward to seeing everyone next month for another one of these.
Okay, thank you. Bye.
Thank you so much.